Okay, guys, let us start our today's session. So, in the last lecture, we were discussing about the meiosis and we were discussing about meiosis first. So, as you now know, meiosis first is divided into four phases: prophase first, metaphase first, anaphase first, and telophase first. In last lecture, we discussed about prophase first. Now, you know, prophase first, it is the longest phase, and this phase is further divided into five phases. I told you yesterday, if you want to remember those five phases easily, then you have to remember the mnemonic Lata Zara Pani Dede LZPGD. So the first phase, L, Lata, that is the leptonema or leptotene. In this leptonema or leptotene, initially the chromosomes were in the form of loose fibers known as chromatin fibers. Then in this leptonema or leptotene, these chromatin fibers, they condensed and chromosomes started appearing as thick rod-like structures. The nuclear membrane nucleolus, they remained intact. However, two centrosomes of the cell, they started migrating towards two opposite poles of the cell. So the organization of the spindle that started in the leptonema or leptotene. Then the cell progressed into the zygonema or zygotene. In zygonema or zygotene, homologous chromosomes, they formed pairs. The chromosomes settle are condensing, they are becoming thick and thick. And homologous chromosomes, they formed pairs, right? These homologous chromosomes, in the form of a pair, now they appear as thick rod-like structures with their centromeres clearly visible. So when two homologous chromosomes form pairs, they are known as bivalents. They are known as bivalents. And these chromosomes are further condensing. They are becoming thicker and thicker. And their centromeres become clearly visible in this zygonema or zygotene. Centrosomes, they are further moving towards the two opposite poles of the cell. They are organizing the spindle. Then the cell entered into third phase, that is pachynemia or pachytene. In pachynemia and pachytene, further coiling of the chromosomes occurred. And the two sister chromatids of a homologous chromosome, they became visible. So far, the homologous chromosomes, they were in the form of bivalents. They had only centromeres visible. This was one homologous chromosome. This was another homologous chromosome. They had their centromeres visible, but sister chromatids were not visible in zygony, right? Now in pachynemia, the sister chromatids of these homologous chromosomes, they are now visible. They are now visible, right? So these homologous chromosomes, whose sister chromatids were not visible, they were known as bivalents. They were known as bivalents. Now in pachynemia, their sister chromatids are visible. And we can clearly see that there are four sister chromatids in these two homologous chromosomes. Now we call them as tetravalents. We call them as tetravalents. And you can see now in this diagram that non-sister chromatids of these homologous chromosomes, they overlap over each other at many places. And this point of overlapping, this is known as chiasmata. Chiasma, and they overlap at many places, so they are known as chiasmata. What happens on these chiasmata? There occurs a process known as nicking and annealing. So chromosomes, they cut and rejoin. So that leads to exchange of gene segments. That is the cause of variation in sexually reproducing organisms, right? So this happens in pachynemia. Then this pachynemia is followed by diplonema or diplotene. In diplonema or diplotene, these homologous chromosomes, they start moving away from each other. They start moving away from each other. And the centrosomes, they have almost reached to two opposite poles of the cell. So centrosomes are continuing their spindle formation. We have discussed it yesterday. Then in the last phase, that is the diplotene. Sorry, that is dikinesis phase. Last phase of prophase first, that is known as dikinesis phase. In dikinesis phase, The centrosomes, they have completed the formation of spindle, nuclear membrane and nucleolus of the cell that disappears. And these homologous chromosomes, they start aligning at the equator of the cell. So these events occurred in the prophase first of meiosis first. So they were the main events of prophase first of meiosis first. Now today we are going to discuss about the metaphase first of meiosis first. So we will see what happens in the metaphase of this meiosis first. Now in metaphase, cell leaves the prophase first and enters into metaphase first. In metaphase first, the spindle formation is completed already. The homologous chromosomes, they are perfectly aligned at the equator of the cell. Homologous chromosomes are perfectly aligned at the equator of the spindle of the cell. So these are the homologous chromosomes. They are perfectly aligned at the equator of the spindle of the cell. But they are perfectly aligned around the equator of the spindle. So in case of human cells, all the 23 pairs of chromosomes, they are perfectly aligned around the equator of the spindle, right? So at this metaphase stage, the chromosomes are most highly condensed. They are most highly chromosomes are most highly condensed, and they are perfectly aligned around the equator of this spindle. So this happens in the metaphase first. Then this metaphase first, it leads into anaphase first. What happens in anaphase first? I will show you that in diagram. Homologous chromosomes, they are separated from each other completely, and they are being pulled towards the two opposite poles of the cell by kinetochore spindle fibers. So as you must be remembering, these chromosomes, they are attached to kinetochore spindle fibers. These kinetochore spindle fibers are pulling the chromosomes to this side. These are pulling the chromosomes to this pole. And these kinetochore spindle fibers, they are pulling the chromosomes to opposite pole of the cell. So in anaphase first, the kinetochore spindle fibers, they shorten. Kinetochore spindle fibers shorten. They shorten by the removal of tubulin dimers at the microtubule organizing center. That means at the centrosomes. So at the centrosomes, tubulin dimers are removed from kinetochore spindle fibers. And these kinetochore spindle fibers shorten. So when they shorten, they pull these homologous chromosomes towards two opposite poles of the cell. So in anaphase, all the 23 pairs of chromosomes, they are separated from each other. So taking 23 chromosomes to this pole and 23 chromosomes to this pole, right? So homologous chromosomes are separated from each other and they are pulled towards the two opposite poles of the cell in anaphase first, right? Then this anaphase first, it is followed by telophase first. So what happens in telophase first? So chromosomes, when they have reached to two opposite poles of the cell during anaphase first, now these chromosomes, they uncoil. Chromosomes uncoil. And they again start appearing as thread-like structures. Then they get surrounded by a nuclear membrane and the nucleolus reappears. Chromosomes get uncoiled. They get surrounded by nuclear membrane and the nucleolus also reappears. So we can say in anaphase first, reconstruction of nuclei. Reconstruction of nuclei. Reconstruction of nuclei. Nuclei occurs in the cell. Occurs in the cell. So in anaphase first, the chromosomes are pulled to two opposite poles of the cell. So at the anaphase first, 23 pairs of chromosomes reach to this pole and 23 pairs of chromosomes reach to this pole of the cell. Now, after anaphase first starts telophase first. Now, all these 23 pairs of chromosomes which were present at this pole, they uncoil. They again become thread-like. So they assume the shape of chromatin fibers. And these chromosomes then get surrounded by a nuclear membrane. And nucleolus also reappears. And these chromosomes which are at this pole of the cell, they also uncoil. They also assume the shape of chromatin fibers. They also get surrounded by a nuclear membrane and the nucleolus reappears, right? So we can say in telophase first, reconstruction of the nuclei occurs at the two opposite poles of the cell. Kinetochore spindle fibers, they disappear. However, non-kinetochore spindle fibers, they are still there. These are non-kinetochore spindle fibers. Non-kinetochore spindle fibers, they are still there, right? So in telophase first, the reconstruction of the nuclei occurs at the two opposite poles of the cell. Now this cell is having 46 chromosomes, but 23 chromosomes are contained in one nucleus at one pole, and 23 chromosomes are contained in another nucleus at another pole. So this nucleus is having 23 chromosomes, and this nucleus is also having 23 chromosomes. So in telophase first, two nuclei are formed within the single cell, right? And reconstruction of the nuclei has occurred. Now this telophase first, it leads to cytokinesis. It leads into cytokinetic phase. It leads into cytokinetic phase or cytokinesis. Cytokinesis. It undergoes cytokinesis. So in cytokinesis, what happens? Now this cell, which has passed the telophase 1 stage, now it will divide into two daughter cells. Now this cell will divide into two daughter cells. How the cell will divide into two daughter cells? This cell will now develop a cleavage furrow in the middle. It will develop a cleavage furrow in the middle. As I have told you in the mitosis, it will develop a cleavage furrow in the middle. These are the nuclei with chromosomes in it. So this cell develops a cleavage furrow in the middle. Actually, some contractile protein filaments, they are laid around the equator of the spindle. If this is the spindle, around this equator of the spindle, some contractile protein filaments are laid. Those contractile protein filaments, they contract and they pull the plasma membrane on all sides of the cell inwards at the center, right? So here these protein filaments, they are pulling the plasma membrane inwards. And Finally, this plasma membrane of one side, it combines with the plasma membrane of another side. So when the plasma membrane is joined, it breaks the cells into two. It divides the parent cell into two cells. So this divides the parent cell into two cells. And at this stage, the spindle fibers, non-kinetophore spindle fibers, they also are broken. They also disrupt, right? So finally, at the end of meiosis first, two daughter cells are produced. And both of these two daughter cells, they are haploid. This is one haploid and this is one haploid cell. So these two daughter cells together, they are referred to as a dyad. They are referred to as dyad. So what is the end product of meiosis first? The end product of meiosis first is two haploid daughter cells, right? So in meiosis first, the chromosome number is reduced to half because 23 chromosomes migrated to one pole, 23 chromosomes migrated to one pole. And these 23 chromosomes in telophase, they form one nucleus. These 23 chromosomes in telophase first, they form another nucleus. So then the cell divides. Then this cell with two nuclei, it divides and it forms two daughter cells, each having 23 chromosomes in its nucleus. So this is having 23 chromosomes and this is having 23 chromosomes. So both these cells are haploid. So that is why we call meiosis first as the reductional division. Because in meiosis first, the chromosome number is reduced to half. A diploid cell is changed into two haploid cells, right? And these two cells together, they are known as a dyad. So at the end of meiosis first, a dyad is produced. Two daughter cells with haploid number of chromosomes is produced. So this happens in the meiosis first. Now both these daughter cells, they will enter into meiosis second. Both of them together will enter into meiosis second. So we will see what happens in the meiosis second. Now meiosis second, like meiosis first, it is also divided into following: prophase second, metaphase second, anaphase second, and telophase second. Telophase second. But before entering from meiosis first into meiosis second, the cells, they take a brief rest, and that brief resting period is known as intermitosis or interkinesis. That is known as interkinesis, or we also call it as intermitosis, right? In this interkinesis phase, which is between meiosis first and meiosis second, this interkinesis phase, it is between meiosis first and meiosis second. In this phase, there is no further DNA replication. No DNA replication. DNA replication has already occurred in interphase, in S phase of interphase of the cell cycle, right? So in interkinesis, the cell may take a brief rest, but in this interkinesis, there is no further replication of DNA because each chromosome is having two sister chromatids. Each chromosome is already having double DNA content, right? So in interkinesis, the cell may take a brief rest, but there is no further replication of DNA. However, these centrosomes, they replicate. Centrosomes, centrosomes replicate so that they will form the spindle in meiosis second, right? So in between meiosis first and meiosis second, there is a brief resting period in animal cells. That brief resting period is known as interkinesis, or it is known as intermitosis, right? In this phase, there is no further replication of DNA because each chromosome is having double DNA content. However, the centrosomes replicate, and these centrosomes they will form the spindle in the meiosis second. The spindle of meiosis first, the spindle of meiosis second is at right angles to the spindle of meiosis first. In meiosis first, I drew the spindle like this. In meiosis first, I was making the spindle like this. This was the spindle of meiosis first. This was meiosis first spindle. In meiosis second, the spindle is perpendicular or at right angles to the spindle of meiosis second, meiosis first. So in meiosis second, the spindle will be formed like this. Spindle will be formed like this. So it will be at right angles to the spindle of meiosis first. So you have to keep it in mind. This is meiosis second spindle. So in meiosis second, the spindle will be at right angles to the spindle of meiosis first. So in the meiosis first, I had shown the spindle like this. In the meiosis second, the spindle will be at right angles to this spindle. So the spindle formation will take place like this, right? Now, this meiosis second, it is further divided into prophase second, metaphase second, anaphase second, and telophase second. Which cells are going to enter into meiosis second? Both these two daughter cells, which were formed at the end of meiosis first, this diode, it will enter into meiosis second. It will enter into meiosis second. So we will see what will happen during this meiosis second. Now this diode, these two daughter cells, which were produced at the end of meiosis first, they will now enter both together into the meiosis second. So both the cells will enter into prophase second. Both the cells will enter into prophase second. So what will happen? What will happen to these cells in prophase second? Each daughter cell had a nucleus. Each daughter cell had a nucleolus in that nucleus, right? Each daughter cell had fine thread-like structures in the nucleus known as chromatin fibers. So I will show it here. So when the cell enters into prophase second of meiosis second, it had a well-developed nucleus in it, covered by double nuclear membrane. It had chromosomes in the form of fine thread-like structures known as chromatin fibers. It had a nucleolus, right? And it had centrosomes. It had a pair of centrosomes. This centrosome, it had replicated in interkinesis, right? So this is the position of cell which enters into the prophase second, right? So two cells will together enter into prophase second. The diode will together enter into prophase second. This is the diode. They are the two cells. So this is the diode. This diode is together entering. This diode is together entering into the prophase second of the meiosis second. So now what will happen in this prophase? Now in this prophase, we will take only one cell. We will show the diagram of only one cell. The events are occurring together in both the cells, right? So in prophase second, the nuclear membrane of the cell that disappears. Centrosomes, they reach to two opposite poles. They start organization of the spindle. They organize the spindle. They organize the spindle. The nuclear membrane and the nucleolus, they disappear. Nuclear membrane and nucleolus disappear. And these chromatin fibers, they coil up. The chromatin fibers, they coil up. And they appear as thick chromosomes. And their sister chromatids become visible. And their sister chromatids become visible. Right? So, in prophase II, like the prophase I, the nuclear membrane of this nucleus, it disappears. Nucleolus also disappears. Chromatin fibers, they condense. And these chromatin fibers, after condensation, they appear as thick rod-like structures with their sister chromatids becoming visible, right? And spindle formation is completed at the end of prophase II. Spindle formation is completed at the end of prophase II. So this is happening in both the cells. Now, so this is the end of prophase. This is late prophase II. This is late prophase II. So I will tell you this meiosis second. It is just like mitosis. This meiosis second is just like mitosis. Now this cell will enter into metaphase second. This cell will enter into metaphase second. So in metaphase second, the chromosomes, they are perfectly aligned at the equator of the cell. These chromosomes are perfectly aligned at the equator of the cell. So all the 23 pairs of chromosomes, they are perfectly aligned at the equator of the spindle. They are perfectly aligned around the equator of the spindle. These chromosomes, they are attached to kinetochore spindle fibers. And non-kinetochore spindle fibers, they run from one pole to another pole of the cell. Right? So this is metaphase second. So we had 23 chromosomes in this cell, 23 chromosomes in this cell. So all these 23 chromosomes, in case of human cells, they are aligned perfectly around the equator of this spindle. And chromosomes get most highly condensed. They become most highly condensed. They are thickest at this, right? Now, after aligning the chromosomes perfectly around the equator of the spindle, now the cell will enter into anaphase second. Now this cell will go into anaphase second. So we will see what will happen in the anaphase second. In anaphase second, the kinetochore spindle fibers, they will shorten. Kinetochore spindle fibers will shorten. And they will pull the chromosomes. Kinetochore spindle fibers will shorten. They will pull the chromosomes to two opposite poles of the cell. Breaking the centromere and separating the sister chromatids from each other. So in anaphase second, the kinetochore spindle fibers, they are shortening and they are pulling the chromosomes towards two opposite poles of the cell. So they generate pressure. When the kinetochore spindle fiber shorten, this kinetochore spindle fiber is pulling the chromosome towards this side. These kinetochore spindle fibers, they are pulling the chromosome towards this side. So when kinetochore spindle fibers generate pressure on the centromere, then the centromere divides and two sister chromatids of these chromosomes, they get separated from each other. They get separated from each other. So 23 chromatids will move towards this pole of the cell, and 23 chromatids will move towards this pole of the cell. So this happens in the anaphase second. So in the anaphase second, the two sister chromatids of a chromosome, they get separated from each other. They get separated from each other. So how they separate from each other? They are separated from each other by the shortening of kinetochore spindle fibers at the two opposite poles of the cell. So when these kinetochore spindle fibers shorten and these kinetochore spindle fibers shorten simultaneously, these kinetochore spindle fibers pull the chromosome towards this side, they pull it towards this side, and this chromosome, it breaks in the middle, separating one sister chromatid from another sister chromatid. So in this way, 23 sister chromatids will reach to this side and 23 sister chromatids will go to this side. So this divides the DNA content of the cell. This divides the DNA content of the chromosomes. This chromosome was so far having double DNA content. In its two sister chromatids, it was having two DNA molecules. Now, when this chromosome breaks in the middle, now every sister chromatid, every chromosome is having a single DNA molecule in it. This happens in the anaphase second. Then what happens in the telophase second? Then what happens in the telophase second? In telophase second, as I told you, there is a reconstruction of the nuclei. These 23 sister chromatids, they will reach to this pole. 23 sister chromatids will reach to this pole at the end of anaphase second. Now in telophase second, all these 23 chromatids, they get surrounded by a nuclear membrane. They get surrounded by a nuclear membrane. Nucleolus reappears. Nucleolus reappears. Kinetochore spindle fibers disintegrate. However, non-kinetochore spindle fibers may be settled here. So in telophase second, there will be again a reconstruction of the nuclei. A reconstruction of nuclei. Nuclei are again reconstructed, right? So in telophase second, nuclei are reconstructed. So now this cell in telophase second, it has got two nuclei in it. Now this telophase second, it is followed by cytokinesis. It is followed by cytokinesis again. Now this cell, it will divide into two daughter cells. This cell will now divide into two daughter cells. So two cells had together entered into meiosis first. So I am showing you the fate of one cell. Same thing happens with another cell. So this cell will divide into two daughter cells. This cell will divide into two daughter cells. Again, this will divide into one daughter cell. This will divide into another daughter cell. Right? So similarly, this cell, it will also divide into two daughter cells in the same manner. So at the end of meiosis second, from two cells, from the dyad, now four cells are together formed. These four cells are known as tetra. So at the end of meiosis second, now from these two daughter cells, from this dyad, a tetrad is formed. So this cell divided into two daughter cells, and this cell also divided into two daughter cells. So at the end of meiosis second, four haploid cells. This will be haploid, this will be haploid, this will be haploid, this will be haploid. So four daughter cells are produced. What is the difference between these two daughter cells and these four daughter cells? These two daughter cells, this dyad, it had half the number of chromosomes. But every chromosome of these daughter cells, they had double DNA content. they had double DNA content. One DNA molecule was present in this chromatid and one DNA molecule was present in this chromatid. So the daughter cells which were produced at the end of meiosis first, they had half the number of chromosomes. But in every chromosome there was double DNA content. Right? So these daughter cells, although they had half the number of chromosomes, but still they were having double DNA. At the end of meiosis second, four daughter cells are produced. But each of these daughter cells, it has a single DNA molecule in its chromosome. So that is why meiosis second is necessary. When the genetic material is reduced to half in the meiosis first, then what was the need of, when the chromosome number is reduced to half in meiosis first, then what was the need of meiosis second? In meiosis first, the chromosome number is reduced to half. From 46 chromosomes, daughter cells with 23 chromosomes and 23 chromosomes are produced. But each daughter cell has got 23 chromosomes. But in those 23 chromosomes, it has double DNA content. It has two DNA molecules in each of its chromosomes. So in order to divide this DNA, in order to make these chromosomes with only one DNA molecule, these two daughter cells, they undergo meiosis second. So at the end of meiosis second, they produce four haploid daughter cells. They produce a tetrad. And in every cell, there are 23 chromosomes and every chromosome has got single DNA molecule. So here we had 23 chromosomes, but every chromosome had double DNA content in it. It had two DNA molecules. At the end of meiosis second, we have 23 chromosomes and every chromosome is having just one DNA molecule in it. So we call meiosis first as reductional division because in this chromosome number is reduced to half. We call meiosis second as equational division because it distributes this DNA, double DNA content of these two daughter cells equally into among its four daughter cells, right? So that is all about meiosis first and meiosis second. Now you know meiosis it only occurs in germ cells. It is used for formation of gametes. It is used for formation of sperms and formation of ova, right? So that is all about meiosis. Thank you for being with me.